Hello to all my dear students. I hope you all are doing fine. So today we are going to start with the next lecture of the chapter Molecular Basis of Inheritance, and that is on translation. So far beta you have seen that how DNA is copied to obtain DNA via replication. Then you have seen the mechanism of transcription, that from where DNA is used to obtain the chains of RNA, different types of RNA. And then now we are going to see that how these different types of RNA are used for the mechanism of translation. So basically translation simply means copying of RNA or production of proteins from the chain of RNA with the help of certain enzymes, with the help of different types of RNA in the cytoplasm of the cell. Now be it prokaryote or eukaryote, translation will take place in the cytoplasm of the cell with the help of different types of RNA and of course ribosomal units. So today we are going to see the mechanism of protein synthesis finally. Now, like in beta transcription, the whole transcription process was divided into three phases. Initiation, then comes your elongation, and then finally your termination. Similarly, translation process can also be divided into three stages as initiation, elongation, and termination. So this whole process of translation can also be divided into three steps. Initiation, elongation, and termination. But before we initiate the process of translation, the first step is activation of amino acid. The first step is activation of amino acid. Right? Now, basically beta what happens, there are certain requirements for translation that I should tell you. mRNA chain is required, ribosomal unit is required, magnesium ion concentration is required so that the two ribosomal assembly can be brought together. Energy in the form of GTP is required. Different types of tRNA and amino acids are required for the whole process of translation. Now, सबसे पहले we are going to start with activation of amino acid. That means amino acid 1, which is usually formilated methionine in case of prokaryotes and only methionine in case of eukaryote. So amino acids, before they enter into the process of translation, they first need to get activated so that they can be attached to their specific tRNA for the process of translation. So सबसे पहले you are having an amino acid, be it the first amino acid, which in the presence of the enzyme amino acyl synthetase, that is represented by symbol E, correct, plus energy in the form of ATP. Now what will happen? This enzyme is going to catalyze the activation of amino acid to form an intermediate complex called amino acid AMP. AMP, that means two bonds of adenine has been broken down. This is adenine triphosphate. So two phosphate bonds, they break down to produce adenosine monophosphate. Enzyme E gets attached and inorganic pyrophosphate is released. Now this is the activated form of amino acid which will be now transferred to the specific tRNA in order to activate that tRNA also. So this is the first step. Second step is beta activation of tRNA. Now how tRNA can be activated? To activate the first tRNA, this first activated amino acid need to be attached to it. So tRNA 1 is made to attach with the activated enzyme. So now what will happen? A new complex will be formed between amino acid and tRNA. Enzyme will be set free and AMP will also be released out. Now this is the first component for the translation process ready. After this much work is done in the cytoplasm. Now the initiation process will start. In initiation, beta, the first step is attachment of, now here I am going to teach you the translation of prokaryote. So whatever terminologies I will be using for prokaryote, but parallelly for eukaryote also the differences I will keep on telling you. Right, so first step is attachment of 30S, the smaller subunit first attaches to the mRNA chain. So attachment of 30S ribosomal unit to the mRNA chain. That means suppose this is mRNA, correct? Polarity 5' to 3'. This mRNA is a chain of number of nucleotides. Basically mRNA is a polymer of nucleotide. Now whole mRNA is not going to undergo translation. The initiation codon is AUG and termination codon can be either UA, UAG or UGA. Now this region which is flanking between the initiation codon and the termination codon, only this much region undergoes translation of mRNA. Otherwise, the region which is left behind 5', this is called as untranslated region before initiation codon. And the region of mRNA which is left after termination codon, this is again known as UTR or untranslated region of the mRNA. Correct? Now, what happens beta? In the untranslated region, prior to initiation codon, there are specific sequences located called Shine-Delgarno sequences in case of prokaryote. So what am I saying? Now, in this region, the upstream region, basically, the upstream region of mRNA is having some specific sequences called Shine-Delgarno sequence. These Shine-Delgarno sequences, they are recognized by the 16S rRNA component of 30S. Now beta, 30S is the smaller subunit of 70S, which is made up of 16S rRNA and some proteins. Right? Now this 16S rRNA of 30S subunit reads the Shine-Delgarno sequences. Why? Because the sequence of 16S rRNA is complementary to the sequence of Shine-Delgarno. So with the help of the 16S rRNA, which is complementary to the Shine-Delgarno sequence, 30S ribosomal unit attaches to the right position. So 30S ribosomal unit is going to attach on the right position with the help of, once again I am telling you, with the help of its 16S rRNA. 16S rRNA has some complementary sequences complementary to Shine-Delgarno located in the upstream region, which it reads and then let 30S ribosome attach at the right location to form 30S plus mRNA, to form 30S mRNA complex. Right? So this much is the first step under initiation. Soon after this beta, as soon as 30S joins, what happens? As soon as 30S ribosome joins on the mRNA with the help of its 16S rRNA, two partial sites get exposed called as P site and A site. Called as P site and A site. P site represents the site which is present for the first codon and A site represents to the codon to the location for the second codon. So now let me draw one more diagram. So the second step in initiation is this. Suppose this is my mRNA chain. Here is my first initiation codon. Here is the second codon. Here is my third codon. And like this, so many codons are located. Finally, here is my termination codon. Okay, so here 30S ribosome unit got joined with the help of its 16S rRNA. Now, as soon as 30S joins on the mRNA, two sites are exposed, P and A. P is the first site having the first codon and A is the another site having the second codon. Soon after this, the activated tRNA, which you activated prior to initiation, that activated tRNA, like this is that structure of tRNA now, that is bringing the first amino acid F-met in case of eukaryote, or in case of prokaryote, and normal methionine in case of eukaryote. So this tRNA1, who is having anticodon UAC, right? AUGK, what are the anticodon UAC? So tRNA having anticodon UAC comes and binds to this position. So now what will happen? The next step of initiation will be AUG triple U. Over here 30S ribosome was there. Now soon after, a new tRNA, this tRNA1 comes and gets attached over here, which is having F-met with the help of anticodons UAC, and a bond is established. So this is the first step. Clear? Now soon after this, soon after the attachment of the tRNA to 30S, the 50S is called upon. The 50S now unit comes to complete the ribosomal assembly. This is still here is initiation. This is initiation. Soon after initiation, now comes the next, that is called as elongation, beta. Now what happens in case of elongation? See, what was the condition? Let me draw it again. First codon, second codon. 30S has been joined, because of which now activated tRNA has come and attached itself to the right location, followed by completing the ribosomal assembly 50S. And now to keep this assembly together, critical concentration of magnesium ion is required, which is parallelly being maintained in the cell. Right? Now, meanwhile, the second amino acid is also getting activated. Now second codon is triple U, which codes for amino acid phenylalanine. So now another amino acid will get activated, which will be phenylalanine. This activated amino acid will bind to its specific tRNA having anticodon AAA, right? U ka complementary is A. So now second activated tRNA having anticodon AAA will come and bind to this position. It is going to bring phenylalanine. Here 3 anticodons will be there, which are going to attach to the codon with the help of hydrogen bond. Now what will happen, beta? The carboxyl group. Now these are amino acids. The general structure of amino acid is having carboxyl group as well as amine group. So these are amino acid. Now the carboxyl group of F-met, right, joins with the amine group of second amino acid. So CH group of F-met and NH2 group of phenylalanine binds with each other. Now here NH2 will come, right? Water will be released. There will be it's a dehydration bond to form CONH like this. So over here the remaining groups will be there, and this bond is known as amide or peptide. Not amide, basically it is known as peptide bond will be produced. So that means these two amino acids will now get attached to each other with the help of peptide bond. Now this peptide bond is catalyzed by It is catalyzed by 23S rRNA of 50S subunit. Now, like 30S is made up of 16S, 50S is also made up of 23S. This 23 acts as a ribozyme. It produces an enzyme called peptidyl transferase that catalyzes the formation of the peptide bond between the first amino acid and the second amino acid. Correct? Now, once the peptide bond is produced, which is CONH bond is produced, now what happens beta? This phenylalanine, which is right now attached to the second tRNA, it detaches from the second tRNA and comes below F-met. Right? Now see, next step will be in the elongation. Okay, and this bond, this formation of bond is GTP dependent. So chain elongation, each chain elongation require 1 GTP. So this bond formation is energy based, energy which is given in the form of GTP. Now what will happen beta? Just a second, let me add one more page. See, so this is a condition. Here is the first codon, here is the second codon, this is 30S, here is the first tRNA attached having F-met. Here is the second tRNA attached, but now the second tRNA amino acid has joined the F-met. It has left its tRNA and it's come below F-met because it has attached to F-met with the help of peptide bond. Now beta, what will happen? Soon after this thing takes place, this ribosome assembly is going to move one step ahead. It is going to elongate in the chain, and now what will happen in the next step beta? Now AUG will be left behind. Now the first triple U will come, and now the next amino acid will be exposed, which may be GUG. So now here is 30S, here is 50S ribosomal, right? Now beta, okay, sorry, wait. Just a second. Where's my razor? Yeah. Yes. So what happens actually? Here was F-met, here was phenylalanine. So phenylalanine will not come, rather F-met will come and will join the phenylalanine. So just change this much little bit. So now F-met has come below PA. PA will not go below F-met, rather F-met will come below PA. Soon after this tRNA will take the exit, and now the one tRNA which was at A site will come at the P site, because now triple U will become P and GUG will now become the A. AUG has, codon has shifted, ribosomal assembly has shifted one step ahead. That means AUG is left behind. Now what was A has become P, and the next codon has become A. So now here one tRNA is already attached, that is having phenylalanine and F-met. Now what will come? Next activated tRNA will come, which will have anticodon with respect to GUG. So tRNA which is having CAC, that tRNA will come and will get attached to the A site. Now GUG beta codes for valine. So this is going to bring valine amino acid. Again the same thing will take place with the help of GTP. A peptide bond will be produced between the carboxyl group of phenylalanine and amine group of valine, catalyzed by peptidyl transferase enzyme that is produced with the help of 23S rRNA. जैसे ही इतना होगा, as soon this work is done, now what will happen beta? Now this PA will detach. Now this PA will come below valine, and again your ribosomal assembly is going to move one step ahead. It is going to continue till the time termination codons are not reached. So now what will happen? See, now AUG is left behind, now triple U is left behind. Now GUG will come at P. Now next codon which may be, for example, AAA, which codes for arginine, will become A. Now here comes your 30S, here comes your 50S. This tRNA, after giving its phenylalanine, will leave the system, and now what will happen in the next step? Here tRNA is attached, which is now having valine attached, phenylalanine attached, and F-met attached. Again, a new tRNA will come, whose anticodons will be GGG, bringing arginine. Again, a peptide bond will be produced, energy requiring step, and this is going to continue till the time termination codons are not reached. So last step is termination. Now what happens, beta? Suppose this is my AAA and UA. So what happens once the ribosomal assembly reaches? Now this will become P site, and this is going to become my A site. Okay, this is the condition. Ribosome is over here, whole tRNA is attached over here, having the first arginine, then valine, then it was phenylalanine, and the first amino acid was F-met. So see, whole chain of amino acid is being produced with the help of mRNA chain. So you can see mRNA is dictating. It is acting like a messenger which tells that which amino acid chain has to be produced. So mRNA is guiding. mRNA is carrying the codon which is going to guide the addition of amino acid in the polypeptide chain. tRNA is helping in bringing the specific amino acid to the specific codon on the mRNA, and rRNAs are catalyzing the process. Peptidyl transferase, catalyst, 23S rRNA. So you can see all the three types of RNA are involved in the process of translation. Now what will happen? Beta, as soon as termination codon comes, now for termination codons, there are no tRNA. So what will happen? Instead of tRNA, some release factor will come, and these release factor, with the help of energy in the form of GTP, they will terminate the process. They will simply, what they will do, as soon as release factor comes and binds to the final codon, to the nonsense or termination codon, they utilize one molecule of GTP and they stop the process by removing the two ribosomal assemblies. The magnesium ion concentration falls below, because of which the two ribosome 30S and 50S, the two ribosomal subunits, they separate. Because of the two ribosomal units separate, this tRNA also gets separated from the codon, mRNA is set free, and later on this peptide chain also breaks and set itself free. So as soon as termination codons come, beta, just a second. Ek second, beta. As soon as the termination codon comes, the two ribosomal assemblies, they separate from each other. 30S, 50S, right? tRNAs are also set free. The tRNAs are also set free. The polypeptide chain is also set free, having all the amino acids which are synthesized. What was the sequence? F-met, phenylalanine, valine, arginine. So whole amino acid chain is also set free, and then release factor also releases themselves free. So this is how the process of translation finally ends. Let me just tell you once again what is happening overall. So first step in translation is activation of amino acid, which is done in the presence of the enzyme aminoacyl synthetase. This activated amino acid is then moving to the tRNA, so that tRNA can also be activated. Then starts the first process, which is initiation. In the presence of 3 initiation factors in case of prokaryote, 30S unit joins to the mRNA with the help of its 16S rRNA component. Now, after that, 2 sites are exposed, P and A. To the P site comes the first activated tRNA, followed by the attachment of the ribosome, thus completing the unit. Now, soon after this, another tRNA respected to next codon comes, and then a peptide bond is produced between the 2 amino acids of the tRNA at P and A site, that is catalyzed with the help of the enzyme peptidyl transferase. Soon after this is GTP dependent. Now this process is going to elongate, and finally it is going to terminate once the termination codons they arrive. As soon as the termination codons arise, there are no tRNAs for them, rather release factor comes and binds to it. With the help of energy, it disassembles the whole setup. The 2 ribosomal units are set free. The tRNAs are set free. Where I have done that? Have I not written? I think I have written somewhere. Oh, the tRNAs are left free, the release factors are released, and everything is done again. Now mRNA chain is also released free, so that this mRNA chain can again be utilized whenever, if suppose same proteins, if same amino acid chain is required, then the same mRNA can again be used for the process of translation. So this is how protein synthesis takes place in 3 steps: activation, initiation, elongation, and termination. It is an energetically expensive phenomena. It requires energy in the form of GTP. The enzyme is ribozyme, which is peptidyl transferase. I hope you all have understood this whole process of translation. If yes, then do not forget to solve these questions. What is the first step of translation? I hope you all can do it. Very easy question. Second, which component of 50S acts as a catalyst? So peptidyl transferase is the enzyme which acts as a catalyst for the peptide bond formation during translation. So which is that enzyme that is acting as catalyst? And it is made up of which component of 50S? I hope you all know these things. You have heard the today's lecture very patiently, and yes, with full attention. If yes, then we are going to keep it till here, simple and short. See you in the next lecture. Bye bye everyone.